In our profession, we understand that the lack of a well-balanced diet has negative consequences on chronic disease. General diet recommendations have been developed for specific diseases, which have been helpful in preventing or delaying the progression of chronic disease to some extent. But why have these diet recommendations failed to produce change in some individuals and succeeded in producing change in others with the same disease? This is the exact question that led to the development of nutritional genomics. Nutritional genomics is a realm of study that focuses on gene-nutrient interactions. These interactions can go both ways. Either the nutrient itself can alter gene expression, or gene expression can alter our body's response to a specific nutrient. By understanding how each individual reacts to specific nutrients, we can enhance patient care and positively accentuate health outcomes. To better understand the complexity of nutritional genomics, let's take a look at the human body. Our body consists of trillions of cells. These trillions of cells each hold 23 chromosome pairs. This means that in each cell there are a total of 46 chromosomes. And in each of the 46 chromosomes, there are hundreds to thousands of genes. And still, it's not that simple, because genes can be expressed in a variety of ways across individuals. What this means in regards to nutrition is that a slight difference in the way someone's genes are expressed might cause an entirely different or even opposing reaction to a specific nutrient. The basis of nutritional genomics involves narrowing in on where these genetic differences are on a chromosome, and even more specifically, which nucleotide pair they associate with within a gene. The variations in nucleotide pairs are known as single nucleotide polymorphisms.
The term single nucleotide polymorphisms, or commonly pronounced as SNPs, are the genetic variations, or polymorphisms, which are present in one's DNA. There are 4 nitrogenous bases which make up our DNA. There's adenine, guanine, cytosine, and thymine, or uracil, when discussing RNA. Each of these nucleotides has a specific partner of which it forms hydrogen bonds with. A bonds with T, or U. G bonds with C, or vice versa. These nucleotides spell out the sequences of our genes. What differentiates you from the person sitting next to you is the arrangement of these single base pairs. Fascinating, right? Well, let's just snip right into it. A SNP occurs when there is variation in a single base pair. So let's break it down. A SNP may replace the base cytosine with the base thymine in a specific stretch of DNA. The switch in nucleotides could either be detrimental or have a neutral effect on one's body, depending on the part of DNA the SNP was located on. SNPs occur every other 300 nucleotides, which means that there are over 10 million polymorphisms in the body. That being said, humans have millions of genetic differences. SNPs are the reasons for differences in appearance, why individuals develop certain diseases, and why certain people respond to different types of drugs. For example, you may have straight hair, and your friend may have curly hair. Or your friend may develop cancer, or they may develop cardiovascular disease. These polymorphisms act as biological markers which help locate genes which are associated with disease as well. Specifically, we're going to discuss further the polymorphisms which occur in individuals who have cardiovascular disease.
One example of a SNP related to cardiovascular disease is found on the ApoA1 gene. The ApoA1 gene codes for a protein that is found in high-density lipoproteins, or HDLs. HDLs are known to be the good cholesterol and are protective against cardiovascular disease. The SNP that occurs on the ApoA1 gene involves a change from guanine to adenine. The A form is considered the rare allele and causes an increased production of ApoA1 protein, which increases HDL formation. So, what does food have to do with all this? Well, it has been found that N3 and N6 polyunsaturated fatty acids influence gene expression differently based on which allele is present. For example, one study compared cholesterol levels in people who were fed a diet rich in polyunsaturated fatty acids. The results of the study showed that LDL cholesterol, or the bad cholesterol, was lowered to a greater extent in women who carried the A allele. No effect was seen in men. Another study showed that as polyunsaturated fatty acids increased in the diet, HDL levels also increased in women who were carriers of the A allele. However, women who were carriers of the G allele experienced the opposite effects. So as polyunsaturated fatty acids increased, HDLs decreased. How can we use this information in medical nutrition therapy? First of all, if they are a carrier of the A allele, increasing their intake of polyunsaturated fatty acids will largely decrease cardiovascular disease risk. If they are a carrier of the G allele, we should suggest decreasing their intake of polyunsaturated fatty acids in order to maintain their HDL cholesterol levels.
Next, we are going to look at another gene which is related to cardiovascular disease. It is called arachidonate 5 lipooxygenase, or abbreviated as ALOX5. This gene plays a key role in the synthesis of leukotrienes, which are mediators of inflammation. Leukotrienes are a part of the eicosanoid family. Eicosanoids are signaling molecules which are derived from polyunsaturated fatty acids, which aid with blood clotting, inflammation, and blood vessel constriction. However, a high amount of eicosanoids in the body can lead to harmful effects. Variation, or polymorphism, in the ALOX5 gene has shown to alter eicosanoid-mediated inflammatory circuits in the arterial wall of the bloodstream, which promotes arteriosclerosis. This is the buildup of plaque in arteries. Those with the variation in the ALOX5 gene have shown to have increased mean intima media thickness as compared to those who do not have the variation. Intima media thickness is the measurement of the innermost layers of the wall of an artery. A study showed that participants with the variation in the gene ALOX5 who consumed omega-6 fatty acids had increased intima media thickness. This is because the omega-6 fatty acids increase the production of eicosanoids, which therefore increases the arthrogenic effect. On the other hand, participants who consumed omega-3 fatty acids showed reduced production of inflammatory leukotrienes and an inhibitory pro-arteriosclerosis effect. Therefore, there was a decrease in intima media thickness. So how can dietary intervention come into play with this gene? Well, omega-6 and omega-3 fatty acids are the determinants which affect eicosanoid synthesis. Consuming omega-6 fatty acids, such as vegetable oils, fast food, and animal meats, increases the presence of leukotrienes. On the other hand, consuming omega-3 fatty acids, such as walnuts, salmon, and mackerel, decreases the presence of leukotrienes. So, to help prevent against cardiovascular disease, it's important that these carriers have an adequate intake of omega-3 fatty acids.
We are going to keep it going with one more gene that is related to cardiovascular disease. This one is called endothelial nitric oxide synthase. But to keep this simple, we're just going to call it eNOS. Of the 3 different variations of eNOS, we are going to focus in on NO. But why is NO so important? NO is going to diffuse from our endothelium to our vascular smooth muscle cells, which compose the wall of the blood vessels. NO is going to increase our concentration of cGMP in our blood. cGMP is a nucleotide that is going to allow for these blood vessel walls to relax and actually increase our blood flow. The release of NO into our smooth muscle found in the wall of the blood vessels is going to regulate our blood flow and also our blood pressure. NO is important because it is going to protect us against thrombocytes from forming together in the body and embedding into our endothelium. But first, a quick refresher on thrombocytes. These are going to be platelets that work to clot our blood. Normal thrombocyte function is important. However, NO is just going to prevent these thrombocytes from actually embedding into our blood vessel walls. This entire process of NO is important for preventing the formation of fatty plaque. So what happens when there's a polymorphism or SNP in the eNOS? This SNP in the eNOS is called GLU-ASP298 and has been associated with ischemic heart disease, which is reduced blood flow to the heart. If this SNP occurs in a patient's genotype, we can recommend dietary supplementation of N3 fatty acids, and we can find these in fish and certain oils. Whereas if our patient's genotype does not have this SNP, there is no association found between the high consumption of N3 fatty acids and reduced cardiovascular disease. So, as you can see, understanding which SNPs a patient has can help us greatly in determining the correct type of recommendation that we can use to be beneficial. As you learn from the different SNPs variations, we can see how complex nutrigenomics is, but also how beneficial it could be to the future of dietetics. The future of dietetics is bright and constantly evolving as new techniques and research comes out. Nutritional genomics is continuously expanding and gaining momentum in the medical field. While nutritional genomics is still new, it can provide light on a new revolution of patient care. It can offer new ways for dietitians to design and also prescribe diets for patients based on their genetics. Giving patients personalized diet care can prevent diseases and also care for current medical diagnoses.